Welcome back. In the second part of this tutorial, we are going to learn about expressing concentration. So concentration basically says that you have a certain volume and then you have steps spread out in that volume. So for instance, let's say that I have a beaker of water and then I put in some salt in that water. I might be interested in the concentration of salt. How could I express it? I might be able to do a percentage. I might be able to do what's called weight per volume. This would be like grams per liter or grams per milliliter. Or I could use something called molarity. So we're going to be looking at some of these ways to express concentration and when they might be used. So let's get started. Concentrations are a concept that most people are inherently familiar with. For instance, if I were to say, you should concentrate your efforts more on a conceptual understanding of physiology rather than trying to memorize it, you would know that that means to spend more time trying to understand physiological processes conceptually rather than spending your time memorizing terms. Here's another example of concentration. Imagine for a moment that I had a room full of students. And these drawings represent an aerial view of the student distribution in each classroom. In the left picture, you would say that the students are not concentrated in any one area, where in the right picture, you would see that students are in fact concentrated in the back left corner of the room. In the human body, we're interested in the concentration of a substance that is dissolved in water. And that is because water is the solvent of living organisms. And of course, that would include humans. So why are we emphasizing concentrations in this lab? In this course, you will learn that molecular transport drives many, if not most, physiological processes. So understanding molecular transport is going to be critical to your ability to understand and conceptualize the physiological processes that you will learn in this class. But understanding molecular transport actually means understanding concentrations, and that is because substances move down their concentration gradient. Now that hopefully is a phrase you've heard before. What it really means is that substances naturally move from an area where they are more concentrated to an area where they are less concentrated. And since most of the substances in our body are going to be dissolved in water, when we think about concentration, we think about the concentration of a substance in water. So now that I've offered a compelling rationale for why concentrations are going to be important in this class, let's dive into how to express concentrations of a substance in a solution. Consider that I have a beaker that is half full with water and I'm dissolving substance diamond in it. Who knows what diamond is? And so I have a concentration of diamond. Now let's say that I draw the same beaker with fewer diamonds. You would say, yes, that is a less concentrated substance. But there's another way that I could make that original beaker less concentrated, and that is simply to keep the same number of diamonds, but expand the amount of water that it's dissolved in. So both of these drawings show a less concentrated solution of diamonds. So what this illustrates is that concentrations of a substance in a liquid are actually a property of two things: the amount of solute and the volume of the solution that is containing that solute. If you change either one of those parameters, you change the concentration. In science, concentration can actually be expressed a variety of different ways. In this lab, we'll cover a few of those different approaches. So perhaps the simplest way to express the concentration of a solution is number of items that are dissolved or suspended in a certain volume of fluid. So if I go back to my beaker example, let's say that the amount of water in that beaker is 1 liter, and I have 6 diamonds suspended in that 1 liter. My concentration would be 6 diamonds per liter of water. Now we most often see this easy approach applied to larger items that are suspended but not necessarily dissolved in fluid. So examples are going to be the cellular elements in blood, most notably red blood cells, white blood cells, and platelets. So when we express the concentration of red blood cells or white blood cells or platelets in your blood, it is usually number of red blood cells or number of white blood cells or number of platelets per millimeter cubed of blood. And we know that millimeter cubed means 1 microliter. The concentration of smaller solutes, such as molecules and ions that are dissolved in the water of the body, are not usually expressed as number of molecules or number of ions per liter of water or per milliliter, but rather expressed by weight or molarity. So let's take a look at those. Let's first look at weight. So if I were to weigh out 1 gram of glucose and dissolve it in 1 liter of water, I would have 1 gram per liter of glucose, and that's my concentration. If I have 3 milligrams of sodium chloride dissolved in 1 milliliter of water, then my concentration is 3 milligrams per milliliter of water. If I were to take 5 milligrams of sodium chloride and dissolve it in 500 milliliters of water, we could actually apply our conversions as shown here. To show that this is equivalent to 10 milligrams per liter. So usually when we do this, we're looking at grams or milligrams, potentially even micrograms. Some hormones are measured in nanograms or picograms even, per microliter of blood or per milliliter of blood, or occasionally per liter of blood. Hemoglobin, for instance, is unusually measured as grams per deciliter. A third way to measure concentration is something called molarity. Now I'm going to put that off for a little bit, and we'll come back to it at the end. And instead I want to focus on a couple example problems that are similar to the ones presented in Lab 1 that look at dealing with the first 2 methods of calculating concentration. The first example problem that I'm going to do is very similar to the red blood cell concentration problem that is presented in your lab. In this case, I'm going to use white blood cells instead of red blood cells, but the concept that you will apply to the red blood cell problem is very similar. Now, red blood cells and white blood cells are very concentrated in your blood. And if you take a drop of blood and you want to determine the concentration, the classic way to do this is simply to count how many red blood cells do you see in this drop of blood. The problem is that, especially for red blood cells, it's really high. So for instance, it's not uncommon to find 5, 6, 7 million red blood cells in a very small drop of blood. That's really beyond our ability to count. So what we would do is we would do a dilution of a sample of blood, and then we would count how many red blood cells are in the diluted sample. So I'm going to give you a white blood cell problem that's kind of similar, and we're going to apply something called a dilution factor in our calculation. So in my example, I am going to assume that I collect 20 microliters of blood from a patient. And I am going to dilute that 20 microliters of blood in 380 microliters of saline solution. Saline solution is just sodium chloride solution that is used to dilute blood. Now, this actually is a 1 in 20 dilution. Here's the math. We take 20 microliters over the total volume of the new diluted solution, which is 380 microliters of saline plus 20 microliters of blood, which gives us 400 microliters on the denominator. So 20 divided by 400 is 1 over 20. So this is a 1 in 20 dilution. And what that means is that the real blood sample, the original blood sample, is actually 20 times more concentrated than this diluted sample that I just made by taking 20 microliters of blood and diluting it in 380 microliters of saline solution. But now what I'm going to do is take a microliter of that diluted sample and smear it on a microscope slide so that I can count the number of white blood cells that I see. Let's say I scan the smeared 1 microliter of diluted sample and I count 373 white blood cells on the microscope. How can I use that information to determine the actual concentration of white blood cells for this person? Since the sample was diluted out 20-fold, we know that the real concentration must be 20 times more concentrated than the number of cells that I observed on the slide. So if I observed 373 white blood cells per microliter of solution, then I'm going to multiply that by what is called my dilution factor. In this case, that would be 20. So 373 white blood cells per microliter times 20 equals 7,460 white blood cells per microliter. But I mentioned in the first tutorial that red blood cell and white blood cell concentrations are rarely expressed per microliter, but often expressed per millimeter cubed. And so we learned that 1 microliter equals 1 millimeter cubed. So the final concentration must be 7,460 white blood cells per millimeter cubed. Now another type of problem asks you to convert from one type of concentration into another. So for instance, let's say that I have a concentration by weight like 10 milligrams per deciliter, and I want to convert that into grams per liter. So how can I do that? Well, it turns out we can use the same techniques that we learned in the first Lab 1 tutorial and apply them here. So let me show you how to solve this problem. Again, I have 10 milligrams per deciliter. I know that I want to get to grams per liter. So what I'm going to do is set up my conversions on the side. One that I'm going to look at is going from deciliters to liters. So I know that I have 10 deciliters equals 1 liter. The other one I need to do is to go from milligrams to grams. So I know that I have 1,000 milligrams equals 1 gram. All right, we're going to start by doing a multiplication sign and a line. And I'm going to put milligrams on the bottom to get rid of milligrams. That allows me to use the second conversion. 1,000 milligrams equals 1 gram. So put 1,000 there, 1 on top. Milligrams cancels out. And now I have grams per deciliter. But I don't want grams per deciliter, I want grams per liter. So I'm going to do a multiplication sign and a line. Now so far we've always put what we're trying to cancel out on the bottom. But in this case, what I want to cancel out is already on the denominator. So I'm actually going to put deciliter on the top. So now that I've done that, That allows me to cancel out deciliters, so I'm going to put 10 deciliters on the top and 1 liter on the bottom. And now I'm where I want to be. I have grams per liter. I'll multiply across the top. I have 10 times 1 times 10. That's 100 grams divided by 1,000 liters. And of course, that's just 100 over 1,000. That's 1 over 10, or 0.1 grams per liter. You can use this technique to solve the problem where it asks you to convert hemoglobin into grams per liter. Now, the final approach to concentration is perhaps the most challenging one for students to grasp. It feels very awkward, especially if you've never really worked with this before, and that is moles and molarity. So we're going to get started by first looking at something called molecular weight. If you've looked at a periodic table before, you'll note that a periodic table, of course, is broken up into various elements, and each element has a chemical symbol associated with it. For instance, hydrogen is H, carbon is C, oxygen is O. And when you look at that periodic table, there are 2 numbers also associated with each element. The upper number is called the atomic number, and that is going to be the number of protons that is found in that element. The bottom number is the molecular weight of that particular atom, or the average molecular weight. So for instance, here I have hydrogen, carbon, and oxygen. You can see that the bottom numbers are the ones I care about. The molecular weight for a hydrogen atom is roughly 1. The molecular weight of a carbon atom is roughly 12. And the molecular weight of an oxygen atom is roughly 16. We don't need to get into too many significant figures here. It would be enough probably for your instructor to just go out to 1 decimal place. So what is the unit of that molecular weight? Well, the unit is Daltons. Daltons is kind of a placeholder unit. We're not really going to use it, but we do want to have something there so that it's not unitless. Now let's get some practice calculating the molecular weight of molecules. For instance, if I have hydrogen gas, which is 2 hydrogen atoms joined by a single covalent bond, the molecular weight is 2 Daltons. How did I figure that out? Well, I know that 1 hydrogen atom has a molecular weight of 1, and then the second one must also have a molecular weight of 1, so 1 plus 1 is 2. Let's look at the molecular weight of carbon dioxide. Carbon dioxide has a single carbon atom. The molecular weight of carbon is 12, and then it has 2 oxygen atoms. The molecular weight of oxygen is going to be 16, so it's 12 plus 2 times 16, and that's the molecular weight of carbon dioxide. I would like you to try to figure out the molecular weight of NH3 and C2H4O2. Go ahead and pause the video and give it a shot. Okay, let's quickly solve those problems. We know that NH3 has 1 nitrogen atom, which has a molecular weight of 14, plus 3 hydrogens, each weighing 1 Dalton. So we have 14 plus 3 times 1, that's 17 Daltons. C2H4O2 is going to have 2 carbon atoms, each weighing 12 Daltons, plus 4 hydrogens, each weighing 1 Dalton, plus 2 oxygens, each weighing 16 Daltons. So I have 2 times 12 plus 4 times 1 plus 2 times 16. Solving this out rather quickly gives me 60 Daltons. Okay, why did we learn about molecular weight? Well, I'm going to tell you that the molecular weight of glucose, which is the most common sugar on the planet, it is C6H12O6, is 180 Daltons. You can verify this if you like, or you can just believe me. It is 180 Daltons, and that's glucose. Now, I can order glucose in a big jar. let's say that I do, and I weigh out 180 grams of glucose. Again, that's the same weight in grams as the molecular weight. It turns out that the number of glucose molecules in 180 grams of this particular substance, glucose, is 6.02214 times 10 to the 23rd power. That's how many glucose molecules would be in 180 grams. Interesting. Let's say that I were to take NH3, and I know the molecular weight of NH3 is 17. Let's say I were to weigh out 17 grams of NH3. Well, guess what? I would have 6.02214 times 10 to the 23rd power of NH3 molecules in 17 grams of that substance. So what was elucidated is that if you know the molecular weight of a substance and you weigh out that much in grams of the same substance, it's always going to contain 6.02214 times 10 to the 23rd molecules. And that constant became known as Avogadro's number. And it's huge. It's a huge number. So it's really bulky and difficult to deal with. So we apply a different name to it. We call that giant number a mole. 1 mole, M-O-L-E, like a mole on your face. So the next thing that you're going to want to figure out is how many moles of substance X is in a particular sample. And this is just another way to describe amount. Again, it's too much to say 6.0224 times 10 to the 23. Easier to say 1 mole, but maybe it's 2 moles, maybe it's half a mole. So how can you figure out how many moles you have? Well, if you have the weight of the substance and you know the molecular weight of the molecule in that substance, you can figure out how many moles there are in that batch. So we're going to use some examples to illustrate this. I'm going to set up 3 example problems to solve. The first one is 10 grams of glucose equals how many moles? 1 gram of carbon dioxide equals how many moles? And 3 milligrams of sodium chloride equals how many moles? So let's go ahead and solve these, and hopefully you'll be able to see the strategy. So I'm going to start with the first one. 10 grams of glucose equals how many moles? Well, I'm going to set it up like a typical conversion. I have 10 grams of glucose, multiplication sign, line, grams go at the bottom. But I need a conversion from grams to moles. Well, this is simply the molecular weight. So the rule is the molecular weight is the grams, and that equals to 1 mole. So we know that the molecular weight of glucose is 180. So therefore, the conversion is 180 grams of glucose equals 1 mole. So now that we have this conversion, we can go ahead and plug it in. We'll put 180 grams at the bottom, 1 mole at the top, grams cancel out, and I'm where I want to be, which is moles. So when I calculate this out, I have 10 moles over 180, which reduces down to 1 over 18. If I plug that into my calculator, that is equal to 0.055 moles. Let's try the second problem. 1 gram of carbon dioxide equals how many moles? So in this case, again, we're going to start with 1 gram of carbon dioxide, multiplication sign, line, grams goes at the bottom. So I need a conversion from grams to moles. Now I can't use 180 because that is not the molecular weight of carbon dioxide. Instead, I need to use the molecular weight of carbon dioxide, which is 44. So the conversion that I will apply is 44 grams of carbon dioxide equals 1 mole. Now that I have this conversion, I can go ahead and put 44 down at the denominator, 1 mole at the top, grams cancel out. I have 1 mole of CO2 divided by 44 If I plug this into a calculator, I get 0.0227 moles. Let's take a look at the last problem. We have 3 milligrams of sodium chloride equals how many moles? So once again, I'm going to start with 3 milligrams of sodium chloride, multiplication sign, line, milligrams goes on the bottom. Now, I know that the molecular weight of sodium chloride is 58.5. So 58.5 grams of sodium chloride equals 1 mole. But I have milligrams down at the bottom, not grams. So I'm going to need to add in an additional step where I go from milligrams to grams. So recall that 1,000 milligrams equals 1 gram. So I'm going to go ahead and start with that to go from milligrams to grams. I'll put 1,000 down at the bottom, 1 gram at the top, milligrams cancels out, and now I'm in grams. So I don't want to be in grams, I'd like to be in moles. So I'm going to do another multiplication sign, a line, and grams goes at the bottom. So now I can plug in my grams to mole conversion. So I'm going to put 58.5 grams at the bottom, 1 mole at the top, grams cancels out, and I am left with 3 times 1 times 1 moles at the top, and 58.5 times 1,000 on the bottom. Now if you plug this into a calculator, you're going to get this really small number, like 0.0000512 moles. It's very common in physiology to have numbers that small. And so once again, we can change our perspective in order to make the numbers a little bit more manageable and meaningful. So often we will not express something in moles, but rather millimoles. Remember, milli just means 1/1000th. So if I want to convert this problem into how many millimoles is this, I simply am going to add on an additional conversion where I know that 1,000 millimoles, and notice the lowercase m for milli, equals 1 mole. So I'm going to take my answer, multiplication sign, line, moles goes on the bottom, 1,000 goes on the top, and now I can solve for millimoles. Now that we have familiarized ourselves with moles, we are ready to learn about perhaps the most commonly used expression of concentration in human physiology, and that is molarity. So molarity is a true expression of concentration in that it is an expression of number of moles of a particular substance per liter of fluid. So for instance, if I have a beaker with 1 liter of water and I add 58.5 grams of sodium chloride to that beaker, I know that 58.5 grams of sodium chloride is 1 mole. So my concentration is 1 mole in 1 liter of fluid. By definition, this is a 1 molar solution, and we use a capital M to denote molar. What if I were to divide the amount of sodium chloride that I add in half? So instead of adding 58.5 grams, I'm going to add 29.25 grams of sodium chloride. Well, technically, I would only be adding half a mole, or 0.5 moles. Still dissolved in 1 liter, my concentration is no longer 1 molar, but rather 0.5 molar. Now, what if I were to say, no, I want to add 29.25 grams of sodium chloride in half a liter? In this case, I know that 29.25 grams of sodium chloride is half a mole, and I'm dissolving it in half a liter. So I have 0.5 moles divided by 0.5 liters, which is 1 molar. So the concentration of the bottom beaker and the top beaker are the same. They are both 1 molar solutions. So we are going to finish with 2 practice problems. I'm going to give you a rather easy one And then I'm going to do the more difficult one that is shown on the last page of your lab. So the easy one is I'm going to assume that we will dissolve 18 grams of glucose in half a liter of water. What is the molarity of this solution? So in order to set this up, I start with 18 grams divided by 0.5 liters. Now, to get to molarity, I need to end up with moles per liter, not grams per liter. So I'm going to use my conversion factor of grams to moles. Again, this is invoking the molecular weight of glucose, which I have already said is 180 daltons. So I have 180 grams of glucose equals 1 mole. And now I'm going to use that conversion factor in this problem. So I'll do a multiplication sign, line, and grams are going to go at the bottom. I'm going to go ahead and put 180 down there and 1 mole at the top. And when I factor all of this out, I have 1 over 5 or 0.2 molars. So again, moles per liter is equal to a capital M or molar. That's the concentration. Okay, the more challenging problem is actually presented in 2 different steps in your lab. I'm going to go ahead and do it all together. This problem says I have 6 milligrams of sodium chloride and I'm dissolving it in 100 milliliters of water. What is the molarity? Okay, so let's start with what I know. I have 6 milligrams divided by 100 milliliters, and I know I need to get to moles per liter in order to have molarity. So the first thing I'm going to do is put up my conversion for the molecular weight. I know that for sodium chloride, there are 58.5 grams equals 1 mole. Okay, so now that I have that off to the side, let me get started. I'm going to do a multiplication sign, a line, and I'm going to start with milligrams and put milligrams down at the bottom. But my 58.5 grams equals 1 mole conversion is not milligrams, so I need to go from milligrams to grams. So I'm going to remind you that there are 1,000 milligrams in 1 gram. So I'm going to plug that conversion in. I'll put 1,000 down at the bottom, 1 gram at the top, milligrams cancel out, and now I have grams per milliliter. But again, I want moles per liter. So I'm going to finish dealing with the grams to get to moles. So I'm going to do another multiplication sign, a line, and put grams down at the bottom. Now I can plug in my molecular weight conversion. I have 58.5 grams on the bottom, 1 mole on the top, grams cancel out, and now I am in moles per milliliter. But in order to express molarity, I need to be in moles per liter. So once again, I need to get out of milliliters and get into liters. Remember, there are 1,000 milliliters in 1 liter. So another multiplication sign, a line, and this time to cancel out the milliliters on the bottom, I need to put the milliliters on the top. I'm going to have 1,000 on the top, 1 liter on the bottom, my milliliters cancel out, and I am finally at moles per liter. If you go ahead and multiply all of this out, you'll have 6 on the top. I'm canceling out the thousands here. So you'll have 6 on the top and 58.5 times 100 on the bottom, and that equals 0.00102 moles per liter, or molarity. Okay, that concludes our tutorial. Hopefully this is enough information for you to be able to finish Lab 1.